Yes, hello students. This recording is for Biology 1406, and this is a walk through the PowerPoint from the OpenStax textbook, Chapter 4, Cell Structure, and we'll also take a look at the study guide for Chapter 4, Cell Structure. So you should know that a cell is the irreducible smallest part of living organisms. So all living things, everything that's defined as alive on Earth, consists of one or more cells, right? So a cell is the smallest living entity. And you're familiar with lots and lots of single-celled organisms already from laboratory. You've looked at single-celled algae, single-celled fungi, that's yeast, and single-celled bacteria, E. coli as an example. And here we're looking at a bunch of cells. This is some tissue, probably from the kidney in an animal, in a mammal probably. These are cells from a plant, from a photosynthetic organism. These may not be green cells right here, but it's definitely a plant because of the kind of brick-like nature of the cells, a very thick, heavy cell wall around them. While these animal cells are more blob-like because they're not surrounded by cell walls, so their shape is not nearly as fixed, and it's even changeable over time. And hallmark of animals and plants is a terrific variety of cells for different functions. And then here are some bacterial cells doing whatever they're doing. All right. So when we look at multicellular organisms, obligatorily multicellular organisms, so that includes plants and animals and certain parts of fungi, think of a mushroom and stuff like that, then what you're looking at is lots and lots of cells of a particular form grouped together into something called tissue. As it says right here, it's a whole bunch of connected or closely allied cells that have a common function, and because of that they usually look very similar to each other. And then in the case of animals and plants, we take a bunch of different tissues in different ways and put them together to make an organ. All right, so every organ in your body consists of a bunch of connective tissue, perhaps some muscle tissue depending on what the organ is and what it does, nervous tissue almost inevitably, epithelial tissue somewhere on it in most cases. So there's a bunch of different tissues making up an organ. Same thing for a plant. It's always going to be consisting of ground tissue, epidermal tissue, and then vascular, circulatory tissue, vascular tissue, and that will always make up the organs of a plant, like a stem or a root or a leaf or a flower or a fruit, or indeed a seed. And then, carrying on with this idea of, increasing complexity, we put, lots and lots of different organs together to make an organ system, and then that's usually what we refer to when we talk about a multicellular organism. Now, a little bit about these organs and then organ systems. If you look inside an individual cell, which is the subject of this chapter, just a single cell, we're focusing on single-celled organisms in 1406, each eukaryotic cell has what are called organelles. So they are separate membrane-bound compartments inside them that do special jobs, kind of like an organ does in you and I. So it's going to be a particular compartment with a whole bunch of specific molecules, usually closely associated with each other, that form, contribute to a function. And indeed, we've got organelles. Organelle means little tiny organ-like thing, inside cells that are grouped together into organelle systems. The biggest of those, and the one that you do need to know, is what's called the endomembrane system. Endo being inside, membrane being membranes, a whole bunch of different membranes defining compartments, and then a system of those that gets a collective job done in eukaryotic cells. Prokaryotic bacterial cells also do all of this sort of stuff, but at the molecular level without the larger organizations with lots and lots of copies of a given molecule grouped together. They don't tend to be built that way. All right, a little bit about size in cells. So, typically we can't see these with the naked eye. It totally depends on the cell in question. There's plenty that can be seen. just by eye under the correct lighting conditions and the correct contrast. But typically they're small enough that they need magnification in addition to correct lighting and contrast in order to see them. This is an optical microscope, a compound binocular optical microscope like you would be using in lab if we had our physical labs together. This one's got an adapter for a camera to fit on top right here. This is a projecting electron microscope right here. So this beams a pile of electrons through a target and then hits a photodetector plate on the other side of the target. So you can take internal snapshots of very small things with this atomic microscope right here. You're familiar with this idea from laboratory, so magnification and resolving power. Magnification is just the brute force ability to increase the space between beams of light. Resolving power is the ability to discriminate between very, very small things and see that there's a space between them. So, we can see these various structures on the surface of this diatom right here, and all these various features right here. So resolving power is the ability to distinguish between very small things when you have an image magnified. So you can magnify any image, but it might become very, very blurry. In computer talk, we would say pixelated, where we don't have a lot of resolution. But if the pixels are very small, if the magnification, the lenses are really clean, and the wavelengths are carefully controlled, we can, have very high resolving power as well. And that's really what we're after when we want to magnify something. We want to, maintain the ability to resolve very small things from each other, and that's, resolution. All right. So here's a low resolution image, using light microscopy right here. And there's a lot to see on it. This is an organism called a radiolarian. It's a photosynthetic little marine organism. I think there's freshwater varieties as well. It has a hard external test shell, and then an amoeba-like cell lives inside here, and it can stick its little amoeba-like pseudopods out through the holes right here to feed. Here's a very high resolution image of the same thing. Done with an electron microscope. So, we can get much higher resolution with an electron microscope than we can with visible light, and that's why we use these things to look at really small things in great detail. Notice both of them, same magnification, same scale bar, different means of illuminating. All right. So here's some, illustrations of just, what you have to do with light in order, to see cells clearly. Here's a light micrograph right here where the angle of light, the angle in that it was introduced into the slide was changed, and that makes the background of the cell really jump out against, its surroundings, whereas this is what it looked like without, the modification. What's happened is, they've adjusted the diaphragm, raised or lowered it, and they've probably closed down the iris really, really, really tight, and then increased the light intensity to compensate to get this image right here. Here, on the other hand, we've taken these cells and we've stained them with methylene blue, which adds, some substantial contrast to the image and allows them to stand out, whereas they wouldn't otherwise. These would be cheek cells from the inside of your cheek. That's typically what this image here is, flat epithelial cells. All right. So again, electron microscopes allow us to see inside, a very, very small resolution, very, very small resolutions. Here we're looking at a mitochondrion in cross-section. These are all of the internal cristae of that mitochondria. And here we're looking at some red blood cells. You notice that very classic concavity right here. There's no nucleus, no organelles inside a red blood cell. So it's this soft, flexible pancake, almost completely filled up with, cytoplasm. But that cytoplasm consists primarily of hemoglobin molecules and a few other enzymes. Anyway, some, microscopy talk right here. All right, this is a transmission microscope shooting through. This is a scanning one that gives you, this is the equivalent of the dissecting microscope. It gives you great three-dimensional images of the surface of stuff. All right. So again, cells are the basic units of life. All living organisms are made of cells. Every living cell on Earth today, is the descendant of a pre-existing cell. And of course, somewhere along the way that process started, but that's not really the subject of this particular conversation. We want to talk about cells as they are. All right. So what are the common components? Every living cell has a cell membrane, often also called the plasma membrane. Plasma membrane, cell membrane are synonyms. They mean the same thing. We use both terms because they're out there. You'll see them, so you want to pay attention to it. Inside that cell, we've got a salty, protein-rich, complex, water-based liquid called cytoplasm. The liquid itself is called cytosol, and then the very large molecular structure is all suspended in it, not dissolved in it, but suspended in it. If you add all of that together, we call it cytoplasm. All right, and so that's true for any cell, any bacterial cell, and any cell in your body. We're going to have DNA inside that cell. There has to be. All right, there has to be DNA inside that cell. In the case of a bacterial cell, it'll typically be small and circular. If it's a eukaryotic cell, it's going to be large pieces of DNA that are linear, and of course they'll be contained within a compartment. But in a bacteria, they won't be in a compartment. They'll just be in a region which is rich in DNA. And then we have large molecular machines suspended in the cytoplasm, and that's the ribosomes all over the place right here. Notice cell wall isn't on this list of 4 things because not all cells have cell walls. None of yours do, for starters. You've got a heck of a lot of cells with walls on them in you. Most of them are bacteria. The rest would be fungus in or on you. But the cells that are genetically you don't have cell walls because that is a characteristic of animals. Most cells out there, by diversity and just by sheer numbers, are equipped with a cell wall, but that is distinctly not the case with animals, and typically with reproductive cells in other eukaryotes. So even though plant cells have cell walls, their gametes, their eggs and sperm, do not, because otherwise the 2 cells couldn't fuse. All right. So, prokaryotic cells, smaller, simpler, right? They lack those internal membranes as a generalization. There's a few that have some, interesting structures internally, so they are the standouts. But as a rule, prokaryotes lack that internal structure, all right? They typically have a cell wall. Most often that is made of peptidoglycan. It may be complex, a cell wall and a membrane inside and outside of it, like a gram-negative bacterial cell, or they might be simpler, with the cell wall being really the outside of the cell, like a gram-positive bacteria. And to the best of our knowledge, earliest life on Earth, and perhaps the first life on Earth, would have been a lot like the bacterial cells we see around today, either archaean or eubacteria. We can put the word EU on here, although when you just write down bacteria, it's implicit that you're talking about eubacteria, especially if you also include archaeans as a separate group here. But archaebacteria and eubacteria are both prokaryotes. And of course, these are the ones that are most likely the most similar to what probably first lived on Earth, the archaebacteria, as in old. All right. So a little bit about bacterial structure. So please get this, correct, from outside to inside, as we work our way through. We're going to have a gelatinous capsule on almost all bacteria. This is what allows them to stick to stuff. So when you fix a slide to a bacterial smear to a glass slide, it's the capsule, which is a jelly-like substance that allows it to glue onto the glass. So all bacteria have some amount of this capsule. Inside of that will be the cell wall, much stronger, structurally substantial. This is what mechanically protects the cell from a lot of penetration and also from bursting due to hydrostatic pressure. So bacterial cells can't suck up enough water to rupture, whereas the cells that make up our body, all of them are capable of overinflating and rupturing in their correct environment. Our outer surfaces are waterproofed to keep that from happening. Inside of the cell wall will be the plasma membrane or cell membrane, and inside of that will be cytoplasm. Notice we've got these pili sticking out right here. These are proteinaceous straws that stick out through the cell wall. They are extensions of the cell membrane, sticking out, evaginations of the cell membrane that allow this cell to interact with its environment. in various ways. In this particular case, we've got a flagellum. Not all bacteria have flagellae. Not all bacteria have pili. Some have both. There's just a terrific amount of variation in there. And notice the circular chromosome, all folded up. Take a long piece of thread, tie it together to make a loop, and then wad it up. And that's what bacterial DNA looks like. It'll always be a loop, if you're careful. Okay. Size. Cells can't be infinitely large, and prokaryotic cells are generally quite a bit smaller than eukaryotic cells. And the reason that cells have a maximum size is the phenomenon of surface area to volume ratio. So the smaller a cell is, and let's just say spherical cells in all cases, if you do the math with any particular, units, you can use millimeters, you can use micrometers, you can use yards, you can use miles, whatever units you want to use, and take a small cell and a large cell, that's a sphere, you'll find that the ratio of surface area to volume is greater when the cell is small, and the volume keeps taking over and becoming a bigger number faster than the surface area as we increase the overall dimensions of the cell. And you might say, well, yeah, of course, as the cell gets bigger, the volume gets bigger. But the key is that the volume increases by the cube of the radius, while the surface only increases by the square of the radius. So mathematically, this will grow faster. And that's what limits how big a cell can be, because if there's not enough surface area to provide exchange of materials with the inside of the cell can't function. There's got to be a minimum amount of surface area for each unit of volume. In prokaryotic cells, that's really small. In eukaryotic cells, because of their ability to transport things around, eukaryotic cells can move large amounts of material quickly inside themselves. Bacteria can't. Because of that, eukaryotic cells can get quite a bit larger because they've got this internal transport system. So eukaryotic cells are also limited. They can't be infinitely large, but they can typically be up to 2 orders of magnitude larger than a prokaryotic cell. What do I mean by 2 orders of magnitude? Well, a prokaryotic cell might be 1 micrometer in diameter. A eukaryotic cell, 2 orders of magnitude greater than that, is 100 micrometers in diameter. And those are kind of typical numbers. Those are not unusual numbers at all. So here's an illustration of that volume issue right here. So if the long and short of it is, if we want to make a big animal, we don't make it out of bigger cells, we make it out of more cells. Single-celled animal, small multicellular animal, bigger multicellular animal. We don't make bigger cells. The cells in a hummingbird are roughly the same size. The cells that do a given job are the same size as the equivalent cells in a blue whale. The blue whale is just made out of more cells and a lot more connective tissue, which is relatively cell-free. All right. So there's a Greek mythological creature called a chimera. You might, if you read it, you might say chimera, but it's chimera, CH. And it's a combination of a lion, a snake, an eagle, a horse, I think. That's a few different things, and there's some different variations on that idea. But it's a combination of a bunch of things. This is a chimeric eukaryotic cell, and by that I mean it's unlikely that you're going to find a ciliated eukaryotic cell that also has flagella, right? Or a flagellated cell that also has cilia. That's pretty unlikely. And then the internal parts, most of your eukaryotic cells have all of these bits and pieces, but not all of them. Remember I showed you a red blood cell earlier? Well, that red blood cell lacks almost everything in this picture. All it is a cell membrane, cytoplasm, and then a whole lot of hemoglobin dissolved in that, and some enzymes. They don't even have mitochondria. They certainly don't have a nucleus, no DNA, no endoplasmic reticulum, and all of this stuff. That's all gone. But, so a eukaryotic cell, unlike a prokaryotic cell, is quite a bit more complex internally. We don't have a whole lot of stuff inside this prokaryotic cell right here, this bacteria. We do have a lot more structure internally. Biochemically, enzymes and molecules that do particular jobs, there the differences aren't so great. Most of the things that this cell does in its various compartments and in its various places, nucleus and all of that, and its mitochondria, this cell gets done in some way with the various enzymes, dissolved and placed all over the place in the cytoplasm of the cell. A lot of the enzymes tend to be right underneath the cell membrane because that's where their supplies from the outside world come, and wherever what they're producing may be secreted back to the outside world. So typically a lot of the enzymes are right on the membrane right here. And then there's a whole bunch of others that are close to the DNA associated with expression of DNA and replication of DNA. But so at a biochemical level, that prokaryotic cell and this eukaryotic cell are quite a bit similar. But structurally, they're different. And the big difference is compartments where we pack particular types of work in one location. And then also the transport system inside the cell, which we'll get to in a little bit. I mentioned organelles. So these would be examples of organelles, all these membrane-bound structures, even this flagellum, even the cilia, technically. Depending on who you're talking to, but definitely all of these membrane-bound compartments internally here. And an organelle system consists of the nucleus, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the Golgi apparatus, transport vesicles, and the cell membrane. All of that collected together is an organelle system, and that is the intra-membrane system, the endomembrane system of a eukaryotic cell. There's an example of a cell system, of an organelle system. All right. We've got a whole chapter devoted to cell membranes, and I'll cover that next. But here is a quick look at the plasma membrane or cell membrane of really any cell, bacteria or eukaryotic. This one here is representing a eukaryotic cell, most likely. It could be from E. coli, it could be bacterial. No, this is eukaryotic, and this is an animal because of the cholesterol molecules right here. That's diagnostic. But what all cell membranes have is this double layer of phospholipids, with the hydrophobic, the water-repelling regions facing inwards towards the center of this bilayer, and the water-attracting heads facing outside to the outside world or inside to the aqueous. Remember, it is a water-based solution filling each of your living cells, the cytoplasm. So facing into this aqueous, somewhat salty pH, about 7.3 liquid inside the cell right here. And then we always have lots and lots of membrane proteins associated with the membrane. Transmembrane proteins that allow materials or information to cross the membrane. Integral membranes, peripheral proteins, which are attached to one side or to the other side, usually acting as anchor points, structural, anchor points for various things. Glycolipids and glycoproteins that are involved in cell-to-cell recognition. These are the sorts of things that viruses take advantage of, binding to one of these things to recognize the cell that they can infect, and then inducing the cell to take them on in, whatever the particular details are. As we can see here, cholesterol molecules, which have 4 rings, right? 3 of them are hexose rings, one of them is a pentose ring, and then there's a short fatty acid sequence sticking off of them. That's cholesterol. It contributes to membrane stability, so that's an important feature in animal cells. You won't find cholesterol in plant or fungi, but you will in animals. So this is an animal membrane right here. And then components of the cytoskeleton inside a cell, which are attached to the cell membrane as well. And no bacteria has a cytoskeleton, so this still says we've got a eukaryotic cell. Because of this right here, I know we've got a eukaryotic cell because we have a cytoskeleton, filaments for transport and for holding the shape and changing the shape of the cell on the inside the cell. That's the cytoskeleton. That's a eukaryotic feature. And then cholesterol tells us on top of that, we're also looking at just strictly an animal cell in this particular case. So you want to know a little bit about cell membranes. All right. So going and taking a look at cytoplasm. Again, cytoplasm is primarily an aqueous solution, but it's got a lot of stuff dissolved in it, including a lot of proteins and sugars that hold a lot of water really close to them. So that really makes this stuff more gelatinous. So if you've ever mixed Jell-O and it's just beginning to set, not when it's really hard, but just beginning to set, that's a lot like what the cytoplasm is. It's a fluid, but thick fluid because of all the stuff dissolved in it. Just like honey is a water-based solution, but the sugar molecules hang on to so many water molecules that it turns into this jelly-like stuff that flows slowly. Well, that's kind of cytoplasm. And cells are typically more liquidy towards the center and more thick and jelly-like towards the periphery, as a rule. Here's the nucleus, and y'all all know about the nucleus. Everybody knows that it's where we're going to have DNA. To be a little bit more specific, the nucleus consists of all the chromosomes, our DNA out here, an area that typically does not have a lot of DNA in it, called the nucleolus, and this is where ribosomes are first assembled. This is where we put together ribosomes. They end up being out here. These red dots represent ribosomes on rough ER. We can also have ribosomes just floating in the cytoplasm, but they're produced here. The proteins are made out here in the cytoplasm. They're imported into the nucleus. They're combined with RNA in the nucleus. Now we have a ribosome, and then those ribosomes are taken back out. So it's kind of a complex process. The way in and out of a nucleus is through the nuclear pore, which is like a grommet in the membrane surrounding the nucleus. And notice the nucleus has a double membrane. There's the inner layer here, and then it folds back on itself, and it's got this outer layer right here. And in addition, it has evaginations, which can be really, really quite extensive, and that is the endoplasmic reticulum. Endo meaning inside, plasm, the cytoplasm, reticulum meaning branched. That's what those words mean. So this branched structure inside the cytoplasm, that is a terrific extension of the outer nuclear membrane. There's the inner membrane and the outer membrane. Rivets, not rivets, but grommets punched through that allow traffic in and out of the nucleus. Nucleolus, which is typically DNA-free, and it's where ribosomes, the proteins and RNA are put together to make a ribosome in this location right here. No proteins are synthesized inside the nucleus, but the nucleus is full of proteins that were made out on cytoplasm or made in the ER right here and then imported back into the nucleus. That's how they get there. They're made outside in the cytoplasm and then brought in. All right. So a tad more, about the nuclear envelope. Here again is that double membrane, the inner membrane and the outer membrane. And the outer membrane can have these terrific evaginations, which just means extensions outwards rather than inwards, which is an invagination. And we have these pores, which are composed of a whole bunch of proteins that make a controllable passageway in and out of the nucleus here. These pores are big enough to allow double-stranded DNA plus proteins attached to them to leave the nucleus, for globular proteins to enter the nucleus, but all of that is regulated. There's lots of gatekeeping occurring right here in this nuclear pore. Again, the nucleolus, this internal structure right here, is the site of ribosome assembly. Proteins from the outside world coming back in through the pores, combined with RNA from transcription of RNA genes, genes that lead to a piece of RNA that is not messenger RNA, that doesn't end up making a protein directly. That's ribosomal RNA, that's transfer RNA, and all the other RNAs that aren't messenger RNA. Anyway, that stuff is combined together in the nucleolus to make ribosomes, and those will be exported back out the nuclear pores. No ribosomes operate inside the nucleus, to my knowledge, to anybody's knowledge, I think. It's always a cytoplasmic phenomenon. All right. Let's take a look at those ribosomes in a bit more detail. Each ribosome consists, and you need to know this, of a small part, the small subunit, and a large part, the large subunit. And the two of those together, plus a messenger RNA, is a complete ribosome. And these don't exist in their complete state unless there's some messenger RNA around. Because the way it works is you've got small subunits floating around all over the place, large subunits. These small subunits will attach to a piece of messenger RNA, and when a start codon is on that messenger RNA and it's identified, it'll latch onto a particular spot on the small subunit. We'll bring over a transfer RNA with the very first amino acid. Then all of that together allows the large subunit to attach, and now the ribosome is assembled, and we'll start reading this thing and producing a polypeptide chain A protein. All right. The mitochondrion, please don't write down on a test that this is the powerhouse of the cell. That's not a wrong statement, but it doesn't have any detail. You want to say something college level about this thing, all right? So it is a site for the conversion of energy stored in molecular bonds, all right? Typically glucose, but other substrates can be used as well. And we start breaking apart, not the macromolecule chains, but the individual monomers. We start breaking the covalent bonds making up those monomers. So we take glucose and break it apart and back down into carbon, hydrogen, and oxygen, and we use the energy of those molecular bonds to reform ATP. We store energy in the form of ATP, and that's what a mitochondrion does. And a mitochondrion is a highly specialized prokaryotic cell living inside your cells. All right, that's this inner membrane here. This is the bacterial outer membrane. They don't really have a cell wall because they've given up because they live in a very protected location. But then there's this outer membrane which is derived from the host. So this is the eukaryotic cell membrane surrounding the outside of it, not the eukaryotic cell membrane, but a bit of it. And then inside is prokaryotic cell membrane made by the bacterium itself. And then there's the cytoplasm, which is called matrix, but it is just cytoplasm. The folds are called cristae. They give us a lot of surface area. And there's huge numbers of the enzymes responsible for cellular respiration, covering, or penetrating the inner membrane right here. We know this is a bacterial cell. If we wanted to, we could illustrate there will be a circular chromosome somewhere. It's a prokaryotic cell, a circular chromosome somewhere inside here. They have their own DNA. It's circular, just like other bacteria. So mitochondria really are bacteria that are endosymbiotic. They live inside our cells in a relationship where both living things get a benefit from them. So that symbiosis is called a mutualistic symbiosis because your cells gain ATP, this thing gains sugar and a safe place to live. And that's the idea there. And of course, they're characteristic of pretty much all eukaryotic cells. There's 2 or 3 aquatic, deep sea eukaryotic cells that have no mitochondria that have been recently discovered. They are very much the exception to the rule. All right. Another organelle that we have in most eukaryotic cells are peroxisomes. All right. And the peroxisomes contain hydrogen peroxide and variations of that chemistry. These are used to combine with vesicles full of really toxic compounds, and the hydrogen peroxide neutralizes by rapidly oxidizing molecules, breaking them apart, producing water molecules as a byproduct, and usually breaking down molecules that have very toxic qualities. In addition, we do a lot of fatty acid and amino acid metabolism inside peroxisomes. Peroxisome is surrounded by a double phospholipid bilayer membrane derived from the host cell. All right. Some differences between animal and plant cells, as it says right here. Both have microtubule organizing centers, but animal cells have centrioles. Plant cells do not. They both have an area where microtubules are put together, but only animal cells have centrioles. All right. The larger area is called a centrosome. So all eukaryotic cells have a centrosome, but only animal cells have centrioles within that centrosome. We'll take a look at a picture in just a second. There's the pair of centrioles, and the larger region around it with the microtubules coming from it is going to be the centrosome. All right. Animal cells have a centrosome and lysosomes, but plants do not. They don't have that. They have a mitochondrial organizing center, but they don't have the this is kind of misstated right here. They don't have the centrioles. Plant cells do not have the centrioles. They still have a mitochondrial tubule organizing center. So they do have a centrosome. All right. In addition, lysosomes will only be found in animal cells, not in plant cells. So those are things that animals have and plants do. Animals have centrioles and animals have lysosomes. Plants do not. Plants, on the other hand, routinely have a cell wall. Not all their cells, but as a general rule. They're also going to have chloroplasts additionally inside the cytoplasm. All right. And for terrestrial plants, they will also have a really, really big central vacuole that you will never find in an animal cell. And this is how you can diagnose that you're looking at a terrestrial plant cell if it's got this large central vacuole. All right. So here's an animal cell. Notice the centrioles, the peroxisomes color-coded in green right here, and then everything else, true for all cells. Here we've got this plant cell right here. We've still got the ribosomes. We've got various things, but we don't have the centriole pair. We don't have the centriole pair in here. And additionally, we have this large central vacuole right here. And we have, in addition to the mitochondria, which are usually quite a bit smaller than this compared to the chloroplasts, we have the chloroplasts as well, which are typically bigger. Mitochondria are very small. Chloroplasts pretty big. In this drawing, they're shown to be just about the same size, which is not technically accurate. And then all eukaryotic cells have the endomembrane system, nuclear envelope, outer envelope, endoplasmic reticulums, rough and smooth, Golgi apparatus, transport vesicles. All eukaryotic cells have fibrous proteins that are involved in transport inside them, components of the cytoskeleton, etc., etc. Plastids. Plant cells will have plastids that store pigments or store starch. So they'll have they're closely related to chloroplasts, and they're semi-autonomous bacterial derived organelles inside the plant cell that are derived from a bacterium. The ones that photosynthesize are called chloroplasts. The ones that produce pigments like for flowers or the red skin of an apple are called plastids. And then there are also amyloplasts, which are ones that store starch. And that's strictly something that you'll find in plants. Here are the centrosomes, and then the centrioles, which are these very, very specific structures that are always orthogonal at right angles to each other, that are diagnostic of animal cells. And they're made out of microtubules. The particular pattern is 9 sets of 3 in a ring right here, 9 sets of 3. And then either 1 or 2 or sets of 2 tubules in the inside right here. And the whole thing is a centriole. And these are the point of origin for all the microtubule system within a cell and are very noteworthy during cell division. All right. So again, if you want to make a statement, most living cells have cell walls. Plants, that cell wall's made out of primarily cellulose. Fungi, that cell wall primarily made out of chitin. Eubacteria and archaebacteria, primarily made cell wall, present, primarily made out of peptidoglycan. Animals, no cell wall, no cell wall. What's the purpose of the cell wall? Provide structure, rigid structure to the cell, and most importantly, it makes those cells, so fungal, plant, animal, algae, everything but fungus, plants, algae, a lot of protists, everything but animals, those cells are very resistant to swelling. They can't overinflate with water. That makes them capable of pulling water from their environment. Even under extremely difficult conditions, they can get water out of their environment because they can't burst if they overinflate, and so they can create quite a draw. Animal cells have to expend a much greater amount of energy getting water and then hanging on to water because of the lack of a cell wall. The benefit of not having a cell wall is that animal cells can change shapes. Fungal cells, plant cells, algal cells can't change shape because they're surrounded by a cell wall, so they're fixed in shape. Chloroplasts, like mitochondria, are really a separate organism living inside. They can't live in the outside world. They're totally dependent on living inside their host cell, but they have their own circular DNA somewhere. It's not in this particular drawing, internally. And there's 3 layers of membranes. There's an inner membrane, which is terrifically folded. It's all one membrane, just massively folded. Imagine taking a single sheet and trying to fold it, or a Ziploc bag. Take a big gallon-sized Ziploc bag and fold it like crazy, trying to make all these shapes. And that's what you've got with all these grana, these thylakoids, which is just a whole bunch of grana piled together. Or sorry, the thylakoids are the individual little discs. A whole bunch of them together as a granum. Importantly, this is all one surface. So one Ziploc bag folded up to make all of this. Then that whole folded up Ziploc bag tucked inside a much smaller Ziploc bag that's totally full of this thing, plus some intervening liquid. That's the intermediate membrane, or the inner membrane. And then all of that inside yet a third membrane, the outer membrane. So this is a phospholipid bilayer. There's a little bit of liquid right here. This is a phospholipid bilayer. There's a whole bunch of liquid in here. That's the stroma. It's just cytoplasm, particular name for cytoplasm inside a chloroplast. And then this is a phospholipid bilayer, folded over terrifically, folded like crazy, but it's a compartment again. Inside those thylakoids is a distinct space from the inner space, distinct from the intramembrane space right here. All right. Plant cells have this thing called a central vacuole, which aids in applying pressure to the cell wall. And the way it works is, say we put this plant cell in fresh water. The cytoplasm of the cell is hypertonic to the water around it, and it will draw water in. That's the situation. Well, that's the situation right here. The cytoplasm is hypertonic to the water in the environment around it, typically. The central vacuole is hypertonic even to the cytoplasm. So water that goes into the cytoplasm preferentially goes into the central vacuole, which swells and presses the contents of the cell hard against the cell wall on the inside. And that's the preferred state. Every happy plant cell is somewhat turgid, which means under pressure, because of this phenomenon right here. In an isotonic environment, we'll see that that central vacuole can shrink. The cell is not under pressure anymore. And if we dip this cell into a hypertonic solution, more salty or more sugary out here, water will be actively drawn out of the cell and it will collapse. This same thing will happen to your cells. This same thing will happen to your cells. But if you put your cells in this situation right here, they'll just swell till they burst. You cannot, if you inject a bunch of distilled water into your blood, all of your red blood cells will swell and burst that come in contact with that distilled water, and if enough of that happens, it'll kill you. All right, so the idea of these mitochondria and chloroplasts being distinct, separate individuals is the basis for what's called the endosymbiotic theory on the origin of eukaryotic cells. All right, and so again, each one of those organelles, the chloroplasts and the other plastids, and then the mitochondria in all of eukaryotic cells, all have their own DNA, their own versions of ribosomes, and their own internal biochemistry, and they divide on their own. They divide for their own reasons. They don't divide because the host cell is dividing. So there really are separate cells living inside you, endosymbionts. All right. Now we get to that endomembrane system again. The endomembrane system consists of the nuclear membrane itself, the endoplasmic reticulum, vesicles to communicate between the endoplasmic reticulum and the Golgi apparatus, vesicles to communicate between the Golgi apparatus and the plasma membrane. So the vesicles are the means of transporting stuff back and forth around in here. And then we've got, nuclear, endoplasmic, Golgi, and cell membrane, plasma membrane, all as kind of a direction, all right? And as we see here, the function of the endomembrane system is to modify, package, and transport lipids and proteins to different destinations in the cell or outside of the cell. So here's an illustration of what lysosomes do. So your white blood cells, a whole bunch of them, can eat bacteria. And so what they do is they ingest a bacterial cell. Now it's inside a vesicle. They've invaginated it. And that vesicle can fuse with a lysosome that's full of digestive enzymes right here, lysosome. And the lysosomes, release their digestive enzymes into the now shared space, digest the bacterial cell into its macromolecular components, so a whole bunch of sugars, amino acids, the monomers. It breaks it down into the monomers. This is the same digestive enzymes that you use in your stomach and intestines, but here this is at a cellular level. And now the cell can either get rid of this stuff as waste or use it as raw material to stay alive internally. Cells in your stomach produce vesicles, containing digestive enzymes. They make lysosomes, but those lysosomes themselves fuse with the outer edge of the cell and dump their contents into the stomach or the intestines. And that's where, how you secrete that stuff. Where are those cells? Either the lining of your stomach, the lining of your intestines, or in your pancreas, or where have you. So here's the endoplasmic reticulum. And what's going on with the endoplasmic reticulum is that it has a close association with the Golgi apparatus has a close association with the endoplasmic reticulum right here. So here we're looking at rough ER. It's called rough because it has ribosomes attached to it. We can also have free-floating ribosomes just in the cytoplasm. But the ones that are on the ER right here, when they assemble and produce a protein, the protein is going to do one of two things. It's either going to be dumped free into the inside of the endoplasmic reticulum right here, or it will be attached to the wall of the endoplasmic reticulum. And then eventually we can pinch off a piece of this, make a bubble of it. That's a transport vesicle right there. It's going to move using the cytoskeleton. The cytoskeleton will provide the pathway. All right. And that can fuse with one of these compartments of the Golgi apparatus, and then it becomes a piece of Golgi apparatus. That's where the Golgi apparatus came from. Fuse a whole bunch of these vesicles together, and that's how you make these bags right here. And we can take that protein, add things to it, chop things off of it, modify it in the various chemical compartments of the Golgi apparatus right here, and then eventually we can evaginate, make a transport vesicle again from this side of the Golgi apparatus, and move this to the cell membrane, and that's how we get proteins either built into the cell membrane. That's how they all got there. They're following this pathway. Or if the protein is released into the cytosol of the ER, and therefore the cytosol of the Golgi apparatus, that's how we can secrete an enzyme into the outside world, like digestive enzymes, like toxins that we release, whatever the particular function is. So a cell can produce proteins that it keeps in its cytoplasm. That would be a free ribosome. It can produce proteins that end up being mounted onto a membrane somewhere. These membranes, these membranes, these membranes, any membrane anywhere. And that's what's being illustrated right here. And then the third possibility is we could secrete a protein. So it's dumped into the fluid of these compartments, and when we join one of those fluid-filled compartments to the cell membrane, as in this image right here, we can, see if I can move this out of the way. I don't know if you can see that, but I can. We can open this container up and let the stuff out into the outside world. Notice the polarity. If we have a protein sticking out, facing the outside world, it had to be facing in on the endoplasmic reticulum, because we're turning these things inside out as we move them. So a protein facing the outside world on the outside of a cell would be produced inside the endomembrane system. All right. So let me remind you of that. There are three places where proteins can go on a eukaryotic cell. They can be free-floating in the cytoplasm. That would be produced on a free ribosome. Or the ribosome can be attached to the endoplasmic reticulum, and that gives us two other destinations. All proteins that are attached to a membrane, anywhere, and all proteins that are released from a cell to the outside world outside of that cell. All of those must go through the endoplasmic reticulum and the Golgi apparatus, typically. How much of this stuff do you have in a cell? It totally depends on the job of the cell. Some cells barely have any of this. Some cells have vast amounts of this stuff. It totally depends on the particular cell and its particular function. So there is no one answer. There may be one Golgi apparatus. There may be none. There may be 50 in a cell. It just totally depends. Same thing for the amount and extensiveness of the endoplasmic reticulum. Just totally depends. All right. So here we're showing a a protein that's being mounted on the outside of this cell right here. All right. Right. Protein on the outside right here, penetrating through. Here's some smooth ER. If you want to know where cell membrane itself comes from, it comes from this process right here of joining vesicles made out of phospholipid bilayer to the cell to increase the size of the cell, except instead of coming from rough ER and the Golgi apparatus, just plain old new membrane just comes straight out of smooth ER. What happens on smooth ER is we're making more and more phospholipid bilayer, and we pinch off pieces of this and join this to the cell membrane to grow the cell. That's how a cell grows. And then, of course, more water is imported to inflate the cell. Smooth ER, that's how we get more phospholipid bilayer, and really all the other lipids that a cell uses for various functions. Rough ER, primarily associated with proteins that are either going to be membrane bound or secreted out of the cell. So as it says right here, the functions of the smooth endoplasmic reticulum. We do a fair amount of carbohydrate metabolism in them, lots of lipids, steroid hormones, which are all lipid metabolism and modifications. In some cells, we store a terrific amount of calcium. That's going to be true in muscle cells all over the place. Smooth ER of an egg cell has a huge amount of calcium in it. That's a pretty big deal in a lot of cells. Depending on the cell, like hepatocytes in your liver, a lot of this sort of stuff going on. One of the many jobs of your liver. All right. This is some muscle cells right here. They're super, super modified. And here, the sarcoplasmic reticulum is just a very fancy and specialized version of the more general idea of an endoplasmic reticulum. And then the cytoskeleton is organized to a level that you couldn't even imagine in any other cell to make up all the contractile proteins that are involved in muscle contraction. But really all that is the cytoskeleton, and a terrific amount of endoplasmic reticulum are the specialized bits of a muscle cell, along with some interesting stuff here like these T tubules, which is the outer cell membrane running as a tube through the cell, which is pretty unique thing, unique to muscle cells. All right. So what do the Golgi apparatus provide us? Well, lots of compartments for different sort of chemistries at different pHs, different salinities, different conditions. So they'll have groups of enzymes that are distinct in each one of these compartments to do whatever jobs this cell is doing, involving proteins that it's secreting or proteins that it's mounting on its membranes. Look, here's how we make lysosomes full of lytic enzymes. Those can be secreted out of the cell, like digestive cells in your body, in your pancreas, or a white blood cell is going to hang onto those lysosomes and use them inside the confines of the cell to digest white bacteria that it eats. They can eat bacteria and live off of them. Or you've got these mobile cells living in you that are hunters and hunting down bacteria primarily, also parasites. We see the terms cis and trans show up again. Cis just means facing you, and trans means facing away from you. Just like in the cis and trans fatty acids, having to do with the positions of bonds across a double bond there. All right, the cytoskeleton. No cytoskeleton in prokaryotic cells, but a big deal and present in all eukaryotic cells. In all eukaryotic cells, the cytoskeleton gives the cell shape and allows the cell to move things internally from one location to another and to hold things in locations. All right, and in the case of animals, the cytoskeleton allows the cells to change shape so that the cells can move, and that's how animals move. Right. The cell doesn't move within the organism, but it changes shape, and if a whole bunch of those do that, we can bend a joint, right, and articulate a limb or something like that. So there are 3 bits that you need to know. Going from smallest to largest are the microfilaments, very small filaments made out of individual spherical proteins. These are the microfilaments. All right. Intermediate filaments, these are the ones primarily attached to this joint right here called a desmosome. Intermediate filaments made out of structural proteins. They hold things in position and give cells structural strength. They make it harder for the cell to be ripped apart. The microfilaments are primarily involved in moving things locally at a short distance locally inside a cell. Microtubules, the biggest tubes in yellow right here, are very, very large cytoskeletal components that are involved in moving things long distances and moving large things long distances inside the cell. So the microfilaments and the microtubules are both what we call dynamic, meaning things can move along them. The intermediate filaments are not dynamic. They're just like cables strung around inside the cell to hold it together. Very small, intermediate in size, very big. Micro only because it's a tiny tube compared to a straw, but it's a huge molecular structure within a cell. All right, so there's the microfilaments, these little actin subunits right here. It's always two strands wound around each other. All right. The microtubules, much, much bigger. But there it is right there. And they're hollow. They are literally a hollow tube of globular proteins making up a hollow straw. And again, these things allow things to move. This actin filaments are a major component of your skeletal muscle cytoskeleton, and cardiac and smooth muscle, but all muscles cytoskeleton. Microtubules give you that shape of the cell. They're involved in moving things around when cells divide, and move all their contents to two new locations. It's the microtubules that make all of that possible. All right, lots of motor proteins attached to them, so that's how we can move things within a cell. Cilia and flagella are all in eukaryotic cells are all activated and manipulated by microtubules. Cilia are short, and there's lots and lots of them covering a cell. In some single-celled organisms, they are how that cell swims through its environment. In you and I, we have ciliated epithelia in our respiratory tract, in a few places in our excretory system, and in females along the fallopian tubes, you have lots and lots of ciliated epithelia. And in all cases, that's for moving mucus and whatever's embedded in that mucus along. In your respiratory system, is to clear out your lungs, continuously clear them. That's why you always cough up mucus. In your fallopian tubes, it's to move the egg towards the uterus. Flagella in multicellular organisms are usually only associated with gametes, so in us that'll just be sperm cells. But in a lot of single-celled eukaryotic organisms, all the cells are flagellated, and that's how they swim around in the water, searching for whatever it is they're searching for, light or food or whatever, mates. And the flagella and the cilia of eukaryotic cells always consists of, let's see if I've got an image right here. Let me go back to this picture. Where's that chimera cell I was looking at earlier? Here we go. So the flagella is actually an extension of the cell membrane packed with microtubule structures that allow it to bend and change shape and whip around. Same thing for the cilia. They're just smaller, not nearly so well developed, but they've got microtubules in there that allow them to bend back and forth, either to move liquid across the surface of a tissue, like our respiratory system, or in the case of a single-celled organism, that might be their means of propelling themselves through the water. That would be like a paramecium that's covered in cilia, and that's how they swim or propel themselves through the water. All right. Extracellular structures. All cells have something outside of the cell membrane. For most cells, that includes a cell wall, plants, fungi, bacteria. Okay. In animals, it's a softer, more pliable mixture of filamentous proteins called the extracellular matrix. It also has sticky stuff, so basically a capsule of sticky material on the outside of a living cell. All right. That's the extracellular matrix in an animal cell. collagens, fibrous proteins, glycoproteins, linking proteins. Most of your connective tissues have lots of these things attaching one cell to another. It's the collagen between cells that holds cells together inside your body, as an example. All right. The last topic we want to cover in here is intracellular junction. Intercellular. Intra means within one thing. Inter means between two things or more things. Intercity shuttles, intercellular junctions. So that's connections between two cells. All right. So animals and plants both do this in different ways for different purposes. Generally, it allows direct chemical communication between two adjacent cells. All right. So here we're looking at plasmodesmata, which is a plant characteristic, which is why we've got the green right here. It's not technically accurate. The cell wall is in fact clear, not green, but whatever. But we've got two plant cells right here with a very large opening connecting the cytoplasm of one plant cell to the cytoplasm of another plant cell. By the way, how can I tell that these are plant cells? The green is a clue. The central vacuole is the dead giveaway. Plant cell. And plasmodesmata are characteristic of plants. The cells with order and with restrictions are all cross-connected to each other, so cytoplasm is shared from one cell to another. That is thematically standard in plants. All right. In animals, and then how to, the plant cells themselves are attached to each other by virtue of the cell walls sticking one plant cell to another. So how a plant is held together is their cell walls are held together. Okay. In animals, we've got to do something a little bit different. In your epithelium, the outside skin facing the outside world, either in your mouth, on the surface of your arm, inside your digestive tract, on the inner surface of your lungs, all of those epithelia, the individual cells are tightly bound to each other with what are called tight junctions, which are like rivets. It's a proteinaceous plug that crosses the phospholipid bilayer of one cell, the phospholipid bilayer of another cell, and links them together. And then there's a whole bunch of those running around, keeping the two sets of cells closely attached to each other so that we can make a watertight layer of continuous cells where the only way to get into what's underneath this epithelium, we can't go between cells, we have to go through the cytoplasm, the living cytoplasm of cells, and these cells have the ability to reject things, to pump things back out, to raise an alarm if something gets in that shouldn't be in there, and stimulate their neighboring cells. That's your immune response. The epithelium is part of that armor, protecting your insides from the outside world through these tight junctions, attaching one cell to its neighbor. Desmosomes is a tight junction on steroids, giving you a much stronger attachment between two adjacent cells. And what happens is this is a structural protein attached to a bunch of intermediate filaments right here. And these intermediate filaments run through the cell and attach to a desmosome over on the other side of the cell. And that's what keeps your skin from ripping until you pull beyond a certain power. That's what allows your skin to stretch a little bit but not rip. Ultimately, you're pulling tight on all these desmosomes and pulling tight on all these intermediate filaments. And if you pull hard enough, of course, you'll start rupturing all these things as well and tearing that outer layer of skin. And again, I'm talking about the epithelium. I'm not talking about the connective tissue down below, different sort of way in which that's organized. All right. And then in animal cells that need to really actively communicate with each other chemically quickly, we have these things called gap junctions, which is kind of like a tight junction, but with a hole in it. And they can be in the closed position, they can be open, sometimes they're adjustable. It totally depends on the details. All right. When they're open, it allows primarily electrical charges to move right through from one cell to another cell, and it allows signal hormones and things like that to move from one cell right through to another cell through these gaps. Your heart, all of the contractile cells of your heart are cross-connected to each other via lots and lots of these gap junctions, and those gap junctions are what makes the heart be able to coordinate its contractions. One cell, stimulates its neighbor to contract in a wave that passes through the heart. And that is made possible by these open gap junctions, which are grouped together into groups. that is it. That's the main stuff we want to talk about in this chapter for structure and organization of cells.